Great, good morning, everyone. My name is Ted Tenthoff. I'm a Managing Director and Senior Biotechnology Analyst at Piper Jaffray. And Piper is again pleased to co-host the Cell and Gene Investor Day. And I'd like to thank the Alliance for Regenerative Medicine for the commitment to this space and also for organizing another excellent event. The attendance is fantastic this year. I think this is the fourth time I've been here, and I think the event keeps growing. So thank you all for your interest and for participating. I have the pleasure of moderating our first panel today, which is entitled Cell Therapy Infrastructure and Manufacturing Requirements for Commercialization. Over the course of the last 4 years, since ARM and Piper first started holding this event, we've witnessed impressive progress in the global cell and gene therapy and regenerative medicine industries. New landmark legislation in Japan and Europe have passed in order to accelerate cell therapies and getting new treatments, maybe even potentially cures to patients. As a result, Japan has approved 2 autologous cell therapy products over the last year alone. We've seen significant progress in clinical trials with CAR-Ts in cancer, cell and gene therapies in a variety of diseases, and also new regenerative medicines. Big pharma and big biotech interest is at an all-time high, and despite these challenging financial markets, the sector has really never been better funded. With so much going on in the space, I do believe the timing's perfect for a panel to focus on infrastructure and manufacturing requirements for commercialization. We are at an inflection point as these cell and gene therapies scale in clinical trials and prepare for approval and commercial launch. Now, Edward and Usman and Michelle touched on some of these topics, but we're going to dive a little bit deeper into these issues. And I'm very, very pleased to introduce my panelists this morning. Sitting next to me is Cindy Collins, who's the CEO and General Manager of Purification, Analysis, and Cell Therapy businesses at GE Healthcare. Next to Cindy, I'm pleased to introduce Bruce Levine, who is the Barbara and Edward Netter Professor in Cancer Gene Therapy at my alma mater, University of Pennsylvania. Also with us today is Bob Preti, who's President of PCT, a leading cell therapy manufacturer. And last but not least is David Sourdive, Executive Vice President of Corporate Development at Cellectis. So thank you all very much for joining us. I'm going to start off with a couple prepared questions, and it's a little bit hard to see with the light, but if anyone does have any questions or any comments with respect to the topics that we're discussing, just please raise your hand. We'd like to keep this as interactive as possible. And again, while I'll direct the first questions to individuals, if you have any comments to add, please just jump right in and we'll make this a nice, lively conversation about such an interesting topic. So I'd like to start by asking Cindy to tell us about what GE is doing to help customers scale up manufacturing of cell and gene therapies. And in particular, what's unique about cells versus other biotherapeutics?
Great question. GE is very committed to the cell therapy and gene therapy space and sees this as an opportunity to industrialize the cell processing activities that we heard a little bit about this morning. And when you think about the industrialization of what is needed in this marketplace, we really feel GE is well suited to make that happen. And the analogy I would use is what we've been able to achieve in our bioprocessing business, which is a multi-billion-dollar franchise for GE today. Started a little more than 10 years ago, really with just chromatography media, and has built out wing-to-wing solutions for biologics manufacturing. So we see the same opportunity existing in the cell and gene therapy area. Our initial focus has been in T cells and specifically CAR-T, but we want to build an arsenal of tools that really enable an end-to-end solution that allows for the designing in of our products as well as our competitors' products to create a viable infrastructure that allows robust manufacturing of the cells that is reproducible and reduce as much failure and risk as possible. And within GE, we have an arsenal of technologies, as you might imagine. It's a very diverse company, one of which includes a software platform called Predix, which our chairman talks about quite openly. And we see the ability to bring digital tools as well as instrumentation, consumables, and reagents to the solution of manufacturing cell and gene therapy products. And so this really, we think, allows us to create an ecosystem that will really enable manufacturing, whether that's done at biotech, pharma, or at academic institutions. We want to be there and help move therapies forward.
That's really helpful. Now, Bruce, you run UPenn's CAR-T manufacturing facility. Please tell us a little bit about these operations, and we'll kind of segue from Cindy into sort of what you're doing in terms of manufacturing these cells. What trials are you currently manufacturing product for, and how do you kind of assess yield from the process?
Sure. So we actually started clinical trials in T cells when Carl June and I were at the Naval Medical Research Institute. We had one room in the Naval Hospital. We moved up to the University of Pennsylvania in '99 in a pilot facility. And then in 2005, moved to a GMP facility in the hospital with 10 rooms for doing clinical trials, both in HIV and in cancer at the time. And it was about that time that preclinical work was going on in the facility and the laboratory on the CAR-T project targeting CD19. Now, our programs and our clinical trials were growing throughout the early 2000s, and the hospital and the university saw that and invested about $10–12 million in a facility also in the hospital, doubling our capacity. We then had 20 rooms, 14 of which were cell processing. And it was shortly before that facility opened that we had treated the early patients in chronic lymphocytic leukemia and seen these CAR-T cells kill between 3.5 and almost 8 pounds of tumor in patients that were relapsed or refractory to all other available drugs. And some of you may have seen, we opened up the newest facility. It's actually across the street, dedicated to CARs and oncology with support from Novartis. And that facility brings our total cell processing rooms to about 23. And I give you that history to provide some sense of the exponential growth in our capacity that is paralleled in the number of trials, the number of patients that we're able to treat. And also, I'll mention that about 90% of what we do is T cells, about 90% is in cancer, the remaining is in HIV. We do some dendritic cell trials. We have other projects with other partners, but it mirrors the growth of the field. Currently, we have somewhere between 16 and 18 clinical trials open. Leukemia, lymphoma. We also have trials open in myeloma and in pancreatic, ovarian, and breast cancer. And I think where we were in 1996, we were an orphan field, really very much boutique. There were very few companies in the space providing equipment and tools. And since the past 5 years, I think there really has been an explosion, not only in the investment that you've seen, not only in the academic centers, not only in the companies in this space looking at therapies, but also in the tools companies, whether it's GE or other companies. And that's really what is needed to move this forward, to go from manual to automated process.
Bruce, I want to pick up on something that you mentioned here and ask you to go in a little bit more detail on that, because talking about 23 rooms gives you a sense of the scale that's required to manufacture these products. So what do you see as really the challenges to continuing to increase capacity? How do you plan for long term as we kind of move from, a couple dozen clinical trials to, hundreds of clinical trials or even commercial scale?
Sure. So, and I think Oz alluded to this in the previous panel. We are in generation 1 of this process. It's a manual process, but you look at where biologics were, where antibodies were, and where they are now. So I think the highest priority has got to be not only in the recruiting, hiring, training, but in looking at automation. And we are doing that with Novartis. I'm sure the other groups are doing that as well. And I've made the analogy on this stage and elsewhere that where we are now is the Model T, or maybe even the Model A. And it took some decades to get to the assembly lines that are fully automated. You have people controlling things, but the units, the cars come off the assembly line in much more rapid fashion, and they're much more consistent. And even today, we're seeing smart cars, whether it's a Google self-driving car or a Tesla, cars that will stop automatically when they send something in front. So there will continue to be innovation, and we shouldn't underestimate the pace of that innovation, and we shouldn't be afraid of where things are now, because we know that there will be innovation and improvement.
I like that analogy. Bob, I want to bring you into the conversation. PCT is a leading manufacturer of autologous cell therapies and really have done really scale up into late stage clinical and even product launch. So tell us about where you think the industry is today, sort of in this process of growing to industrialization or a commercial scale. What do you think we've started to get right, and what do we as a field still have to overcome, just specifically from a manufacturing standpoint first?
Yeah, so the perspective that we bring to this is we've been doing it for a long time and have seen these therapies go. You know, I like to tell the story about 30 years ago, scientists saw cells beating in a dish and thought, well, we can build hearts, right? And I don't think anybody realized what journey we were about to embark on. And all of the different expertise and multifaceted disciplines that would need to come to bear to make this happen. I think what we've gotten right now, and it's difficult now, what's really interesting about this panel today and in the last panel, is that it's gotten difficult to be sort of unique, profound around that, right? Everybody, I think what we've gotten right now is that we're no longer saying tomorrow is the day to deal with this issue of deliverability. Because one of the word that came up earlier in the panel of this transformative therapy, the concept, that's really what's driven, put the fuel behind and put the impetus and the mandate for us to now think about how we're going to get these things across the line. I had a conversation years ago when the field was thinking that because of the business models, the mesenchymal stem cells would be the cells that got out to the clinic first, which was kind of opposite, right? It was not that they don't have a role. They have a very, very important role, but it wasn't a business model that would drive that. It was the same thing that was happening within the autologous space. The business models were crushing the potential. And all of that's come together now, and everybody realizes it, everybody sees it. So what are we doing now? There's the recognition that, and back in those days, where I was going with that was David Smith from Lonza had said to me once that it would take, or he said it on a panel, that it would take a building the size of the Empire State Building filled with people and stacks and everything to deliver the types of therapies we were thinking about. And that was really a statement that said, man, we can't do that, right? That's not going to be, and so that field, as well as the field of patient-specific cell therapies, is undergone this complete paradigm shift that we just can't keep going along and building this linearly, because 23, 65, 90 clean rooms, you can build a very good clinical infrastructure, and we've seen that happen in previous companies. You can build a very good, very high quality, very well-trained staff. You can do that, but it's extremely expensive, and it's hard to map that capacity to the actual delivery. So you're either Immunex or you're on the side of having too much capacity. So not enough capacity or too much capacity. The challenge we face, though, is not that thinking. The challenge is, how do we get from here to there? How do we get from the Model T, to the Tesla, if that's the analogy, right? How do we get to that point where we have an automated, I have to think of automation as one solution, but we're in the middle right now of a transition. And we heard on the panel previous to this, we heard this issue of changing processes, potentially changing the product. Those have to be tested clinically still today, because we still, in terms of having a handle on the product, have a much better handle now because we're engineering, what we want to do. So that's transformative in and of itself. but how do we make that bridge? So I think that's, if there's anything that I see happening now is people are looking to see how do you build that bridge, and we see this in 3 hurdles. One of them is get the infrastructure right today for today. Build the right types of clean rooms, train the people, put the GMPs in, and understand that process, and start devolving that process into its units to really understand what it is. The next step is to leverage as much as you can infrastructure. People have money today, they can build facilities, there's academic facilities, there's companies, but not overbuild that infrastructure, because that infrastructure, A, is really difficult to get right in terms of size. And just sizing is one thing, it's people. People are imperfect, right? And it's really hard to run a business with people. We're doing this now for 17 years, and it's not perfect, it's sausage making. And we have to get beyond that sausage making into real solid industrialization. And then the next step is transition. From the planning done in first step, get into that next step where unit operation by unit operation, we change this and end up at the other side with something that's better engineered, better innovated, and you end up being able to deliver it.
I appreciate that. David, the previous speakers touched on allogeneic product and future products, whether they're armored cars or other things. I want to ask you what Cellectis is doing really to move the field from auto to allo, and what does this mean in terms of manufacturing and just the ability to treat more patients, just that transition from away from an autologous product?
Okay. So just a few words about Cellectis. We were founded 16 years ago as a gene editing company, and we're still very strong in that particular field. Actually, some of us have been working in nuclease-based gene editing since the late 80s. We've been working with meganucleases, talons, and other sorts of nucleases, but we now focus on talons because they offer a very broad window between efficacy and possible toxicity, and they allow us to have very strong yields, which is important in the perspective of manufacturing. So today, 100% of our portfolio is about deploying gene editing in adoptive T-cell immunotherapies. What we do is essentially pursue 4 goals when we deploy gene editing in CAR T-cells. The first one is to make T-cells non-alloreactive. That is, to be able to start from healthy donor T-cells and not from a patient's T-cells, and still avoid graft versus host disease by knocking out the natural T-cell receptor in these healthy donor T-cells, manufacture, scale, and have an off-the-shelf frozen pharmaceutical product, which in a way is manufactured like a drug would be, and it's also tested as a drug would be, with some particularities due to the cell therapy component. But gene editing is not just about making allogenic off-the-shelf equivalents or lookalikes from autologous therapies. It's also about changing other attributes in T cells. The second goal we pursue is to make the T cells compatible with standard of care or with combination therapies. Typically, you want to keep the patient under some specific therapy, under something specific, chemotherapy, for example, or antibody therapy that may not necessarily be compatible with your T cells. So you have to engineer those resistance in the T cells. This is what we've done with the first product, making the T cells resistant to alemtuzumab. This is what we're making with future products, making them resistant to fludarabine, and making sure we can continue delivering fludarabine or the products, corticoids, we have the same equivalent in the patients while delivering the cells. It's also a good way to combine therapies. We think addressing solid tumors will benefit a lot from combination therapies. The third goal we're pursuing in deploying gene editing in adoptive T-cell immunotherapies is to actually give the cells capacity to do what they do not do naturally, to be supraphysiological. Tumors thrive because the immune system fails. And if you give more of what fails, it'll fail again. What we want is to make the cells capable of circumventing the defense mechanisms of the tumors. So in the case of liquid tumors, it's about camouflage. So CARs make tumors more visible. Putting a CAR in the case of ALL does a great job, and it's very, very spectacular. In some tumors, like solid tumors, they are already visible. So it's not just about making them more visible with a CAR. There are checkpoint inhibitors, so what we typically do is knock out the PD-1 gene in the T cells, and make these T cells refractory to the naturally occurring defense mechanism in the tumor cells. There are other candidates, other mechanisms of the sort. And the fourth goal we're pursuing in deploying gene editing in CAR T cells is to broaden the spectrum of addressable targets. Some targets are complicated because they happen to be naturally occurring on T cells. So if you make a CAR against those, you'll observe fratricide killing between the cells, especially during manufacturing. So you need to knock out that target first, and then you can make a product. This is typically what we do with our UCART CS1 and UCART CD38 products, where these targets happen to be present on T cells, and so we have to knock out these targets first. The first product we made was part of an alliance with Servier, so now they have taken over the product last year, anticipating their option, and it's in ALL, CLL, it's CD19. What Michel was saying this morning, everybody was going for CD19. The purpose there was really to prove that you could go allogenic, and there was no particular other intention in terms of target. So that's in the clinic now, and they're taking over. But we're pushing our own wholly owned portfolio now with CD123 in AML. We started the GMP activities, and we'll be filing for an IND in the US this year, both in AML, acute myeloid leukemia, but also in a rare indication, blastic plasmacytoid dendritic cell neoplasm, BPDCN, with the MD Anderson. And there will be 3 other products coming up, CS138 and CD22, all allogenic. So what does allogenic mean in terms of manufacturing? Well, it's not about the logistics. It's essentially about the scale. We start from healthy donor T cells, and a donor can only give so many T cells in a single apheresis. For us, it's a starting material that has to abide by standards that we have discussed with the regulatory agencies, and health of the donor, ratios of cells, and so forth. But once it's there, we treat it as a starting material, and within one run, it's about 3 weeks run, we can make hundreds of doses for patients. And the goal, the evolution, we talked about the T model. I think what we're keen on is making series and moving that from the world of graft to the world of industry with systematic products. So it's about, the first goal is to scale up, even further scale up. Then there will be means to further scale up. Today we start from a fourth of a leukapheresis. We could start with a total block and immediately have a gain in efficacy. The second thing would also probably to have an improvement in cost of good. Our cost of good today is in the $15,000 range approximately. We think we can quite rapidly get down below the 5,000 and hopefully in the future at commercial stage even below, which I was touched upon this morning saying, the very first versions of the processes usually are more expensive than the ones downstream. At the end of the day, what we want is patients to be able to walk into a cancer center, not necessarily a highly sophisticated, happy few hospital anywhere, have something off the shelf, even if the patient doesn't have a lot of T cells left in his body, which actually was the case for one compassionate use of our product that was disclosed last year by a British team. It was an 11-month-old infant, and of course she had no T cells left to make an autologous product. So we think having the option of having an off-the-shelf product is a benefit to patient, and also to make it cost-effective and broadly accessible. Our intention is to manufacture that as we did as a pharmaceutical product, and this is the substance of our exchange with the regulatory agencies, is to bring that to the maximum possible extent in that framework, that known framework, with all the normal development and the QCs and everything. Taking into account the reality that the process will evolve and that the product, the definition of the product when it's a cell, is slightly more elaborate.
I want to ask that sort of where David just went to the other panelists, Bob and Bruce in particular, with your expertise. How do you see the scale ultimately going, from clinical trials today to thousands of hematologic cancer patients to ultimately potentially tens, if not even 100,000 patients as we move into solid tumors and other cancers, and even potentially moving beyond oncology at some point? We touched on allo. How does the industry move there? How do you foresee that happening?
Yeah, so a couple points there. One is in relation to what we're doing and our partnership with Novartis. Our role is the first in human new targets, new iterations, improvements, and even piloting automation to reduce the number of operator interventions. And then Novartis in the pivotal and commercial. With respect to the allogeneic approach, and I think David touched on something that is a problem for many of these advanced patients. Their cells are so beat up by the disease, by the chemotherapy. In the case of infant, you can't get so many. So I think allo has a role there. And in the case of this infant, it was a bridge to a subsequent transplant, or for other patients, it may be to other therapy. And touching on what Bob mentioned earlier on the mesenchymal stromal cells, mesenchymal stem cells, whatever nomenclature you want to use, there was a lot of optimism in the scientific community, but maybe even more in the business community, because they thought, oh, off the shelf, that's something we can give to everyone. But there have been problems with the mesenchymal stromal cell trials and in advancing those. So, and I think, look, the immune system is a product of a couple of 100 million years of evolution. And to have an allogeneic product persist long-term and control disease is a very high hurdle. But certainly there is a role in some settings. And I think going forward, there will be a role for the autologous products as well as the allo products.
Okay, great. Cindy or Bob, do you have anything to add to that? Or?
Only what I was saying before. I think, the model, as the model evolves, we try to concentrate on 4 key factors that people have heard me say many times. But the first is sort of robust, high-quality production of cells. And that's really a quality by design issue. That's an issue of training. That's an issue of putting the right staff in. I mean, you can do that in a variety of different ways. But when you start moving into this cost of goods equation and recognize that at scale, the cost is prohibitive at the moment. It's going to be very, very difficult to work that out. Then you've got to address the cost of goods, address the scalability issue, and address the sustainability issue. And there are words that you hear people talk about all the time. But if you knock off all 4 of those and do that in a very organized way, you can actually transform today's process into tomorrow's process without affecting the product at all. But the thought that we can do that by looking at the incoming material and the outgoing material and not really pay attention to the steps within the process on a very specific unit operation way is, I think, where we've tripped up before, because we don't fully understand the full nature. We abdicated the responsibility of these therapies to the cell a long time ago and said whether the drugs have a limited effect. We know exactly what they do kind of right when they do it, when that point of PK/PD point of contact, we know what they do, but the diseases come back. So we said if the drugs don't know what to do, the cells must know what to do. So we threw them in the body, and we abdicated mechanism of action to the cells. That made it very difficult to actually make product development because we really didn't know what we were getting. So we spent a lot of time trying then to understand what the product was. And now, what's beautiful about the industry, what's made it transformative, is we're telling the cells exactly what we want them to do. So we're kind of getting the best of both worlds. So I think we've really hit that inflection point. And that's what's really catapulted the industry, because we're saying the cells know more what to do, but we're going to help them get there and do their thing. So if you keep all of that in mind, not let the science get out of whack with the engineering, and just be really careful not to build this concept of automation. I think is just, it's a really good concept, but I think we can take it a little bit too far. It's going to be as we imagine it anyway. And it's only, it's just sort of one group's opinion. It's the factory of the future is not a machine that produces a product. And these processes are changing, and the innovation to go from first generation to second to third will change so much that you'll end up with a lot of machines sitting there. And I can point to 10 or 12 of them right now that sit there that have already become obsolete, just as the manufacturing facilities of today will be obsolete. We're building beautiful edifices of manufacturing facilities, and they're dinosaurs as they're being built because they're not the model for the future. So it's really a matter of understanding at the elemental pieces of the process. What is each one of those doing? And then it's a mixing and matching of different solutions to create a very flexible, scalable, sustainable future for the industry.
Yeah,
So
and just to build on what Bob's saying, I couldn't agree more. And when we talk about industrialization and creating a suite of tools, it's really to have that flexibility, to have that robustness in the manufacturing, to enable manufacturing to occur in all parts of the industry, as I said, whether that's pharma, biotech, academic institutions, et cetera, but to really provide a means to make it as robust and foolproof, including applying tools like software as well. So that's how we envision the future. I can use another analogy in our bioprocessing business where today we provide to customers in developing markets flex factories and QBios where we can create a ready-made manufacturing facility virtually anywhere in the world. And so we're enabling other countries and other customers to be able to produce their own products as well. And so we see, certainly not in the near future, that being directly applicable, but also something that we could apply to manufacturing gene and cell therapies as well, because the infrastructure really needs to be there, but it also needs to be highly flexible. And I agree, full-scale automation is not at all what we envision the future to be at this point.
Yeah, I think good points to the left of me and to the right of me. And I get asked all the time, when is this going to be available at community hospitals and made right there at the bedside? Well, maybe never, because there may never be one black box where you throw something into and 20 minutes later it comes out. You've got to worry about comparability, you've got to worry about analytics and comparability of analytics. But it's taking those unit operations, improving those, staying flexible, and one thing I would wager on is that manufacturing is going to continue to evolve in our lifetimes and continue to improve.
Yep. So you don't think they'll ever get to the bedside?
Not
Point
while
of care.
Yeah, well, I would say not in our lifetimes.
Maybe not in yours. I'm younger.
Look, you have very high regulatory hurdles with comparability, and we demonstrated comparability when we transferred the process to Novartis. The more manufacturing sites you have, the more challenges there are in comparability, and that's true for the manufacturing as well as the analytics. So it's a big challenge.
In 1957, I think it was, somebody predicted there would be as many as 5 computers in the world and it would change the world. So I'm not sure.
David,
I don't know.
Maybe one way to look back at this aspect, and actually we started allogenic. We think gene editing can also help in the autologous space, of course, but we focused on that one because we figured precisely what Bruce just said. It's going to be a complicated deployment, but also it's a bit like the bone marrow transplant paradigm. It works, it is deployed, but whose business is this? It's the business of the points of care. So we think this is something that needs to be figured out. This autologous therapy, whose business is it going to be? Is it going to be, is the value added going to be made? Is it going to be at the point of care? We think it will be. And that's why it will continue to thrive, because it brings a lot of value to hospitals. And what part is the industry going to take? How much will be there and how much will be here? And this is not very clear yet, I think. There's still a lot of work to be done to understand. And that's why we focused on the allogenic part, but we think it's a little clearer because there are precedents. We know who does what and where we bring what to the field. I think another aspect also is it tells us is that it's more of a process, of a service than it is a product by definition, because the starting material is so different. And the beauty of it is that actually when CAR T is about recapitulating an acute, something that looks like an acute antiviral response, high peak fever, and then you get rid of the antigen, and it's antigen driven. But when we all get the flu, we all respond more or less the same way, except that we don't have the same starting material. Of course not. We don't have the same clones, and it's all different. And it still works. So focusing on having exactly the same material, it may not necessarily be so important. It's important what expands, and we seed patients with very few cells, and they expand. This is really what is happening. When you look at the numbers, it's very small. That's the beauty of T cells. They expand so much. So all this notion of having standardization of autologous therapies with standardized starting material is very complex. If you look at apheresis strategy, they're different. The methods are different. I'm wondering if you can compare to apheresis coming into this centralized facility, coming from different places of the world. How can you standardize this? I think that will be very challenging. So it looks more to us like a service than it is a product today. The autologous piece, the autogenic piece, you can standardize it because you can screen your donors until you have something which is within boundaries that you have defined with the regulatory agencies.
So you don't see the batch from 100 to 100 to 100 having the same challenge then? You think because you can pre-screen that variation out between large batches?
Well,
this is an issue. This was an issue back with the mesenchymal stem cells as well, right? You have a donor can give a lot of cells, but then you have batch-to-batch variability to look at the same way you would have in the patient-specific setting.
I think there's two components. There's what comes from the donor and what is in the batch variation in the art, in the way the CAR T cell batch can be made, the allogenic CAR T cell batch can be made. So there are some variations, of course. They're within boundaries. There are specifications that have to be abided by. But what we know for sure is that the variations between donors that are screened for certain criteria is far smaller than from patients that have undergone very different histories. And the patient gives what the patient can give as cells, so it's extremely variable. So that sort of prompted us to think of the autologous approach more as a service than it as a product.
So you brought up the FDA, and the prior panel started to talk about the excitement at the agency for these new therapies. What is your guys' view from a manufacturing standpoint about sort of where the FDA is in terms of actually making these cells in a standard process, sort of picking up on some of some of David's answer? And I'll just sort of open that up to really anyone who.
Well, I guess, are you asking what the FDA's view is of manufacturing in any specific way, or?
Yeah, so really sort of where are they in that process? And I know that they've been, again, very engaging with the companies in terms of the clinical side. Is that the same process that's going on the manufacturing side?
Absolutely. There has been wonderful interaction with the FDA, and it's really a dialogue on understanding what the FDA's expectations are, what are the regulations in place, what are the guidances that go along with that, and then recognizing that those regulations and guidances came out and were written for other types of therapies that were not autologous cell therapies. So there are a lot of limitations there, and it's looking at everything from rapid microbiological testing for patients who are in dire need, and how do you define a potency assay, and how do you determine what a dose is for a dividing drug. Okay, so if we give several hundred million cells to one patient, and we give several hundred million patient cells to patient B, let's say, in patient B, they divide and they kill the tumor, but patient A, they don't. Is that an equivalent dose that you gave? Even if you're normalizing to the transduced CAR cells, let's say. So it's recognizing the limits of where we are with the regulations and working together with the FDA on how we think about these things that are not addressed in the existing regulations.
Yep, interesting.
I'd like to add something to that. But, I've had the experience many years ago of interacting with FDA over the devices such as CellPro's device and Baxter's device, the iSelect, many, many years ago, and Cindy remembers these interactions well. And at the time, I was very pleasantly surprised at that time with the interaction, because I had heard, and I had no interaction with CEDAR actually before, but I had heard that dealing with FDA was very, very difficult. The interactions we had were more with CBER. There were some device people involved, but the Office of Cells and Gene Therapy came up a bit later, and it's been an amazingly interactive discussion, right? I think the regulators are struggling the same way the industry is struggling in applying standard drug design methodology to these cells, probably a lot because of the characterization issues and the complexity of the products, the heterogeneous nature of them and all of that, which is famously talked about, and we don't have to go into it here. But I think what we've seen more lately as companies are getting closer and closer to the finish line, the standards do have to increase now and become more like drug development standards, right? And so we're struggling a little bit more. It was very easy early with the Office of Cell and Gene Therapy because really the question was, are these safe? Are the therapies going to hurt anybody? And as long as they didn't hurt anybody, we could go and do the investigations. And as long as we came up with a mechanism of action that was a putative mechanism of action, as long as we tied that back to some reasonable functional assay that spoke to the putative mechanism of action, and if we had a good snapshot assay that kind of spoke to that functional assay, that was a story that was good enough for FDA to say, okay, from a regulatory standpoint, you've checked this box. From an efficacy standpoint, you may not have. And that's the danger, right? So you move along the regulatory pathway thinking you have a product, you're changing it, it's the same, but really from a clinical perspective, it may be shifting off its axis. So I think what FDA has done is really helped us think, and their frustration is, and they've expressed this to us as we go down there with the Alliance and have spoken with FDA about, how can we help? They say, just listen to us. We tell you things and you're not listening. And I think part of that is the immaturity of the organization, the youngness of this organization and of this industry. So as this sector matures, and our ability, we get people, Oz mentioned this before, you bring in companies like Novartis, you bring in the Pfizers, bring in GSKs, et cetera. They have a different ear. They hear the FDA differently. Maybe overhear a little bit, right? But they're hearing what the message will be down the road. So that is going to continue to evolve, but FDA has been, I think, has been a great partner in the industry.
The only thing I would add to that, and I think both Bruce and Bob's perspective is from the clinical and product development side of this, is the question going back to capacity. And is there a path forward where there can be this decentralized manufacturing or this distributed manufacturing to deal with capacity issues? Because ultimately, someone will own the BLA or the regulatory file, but then how do we try to enable manufacturing at other locations, or will we be able to enable manufacturing at other locations? And I think from an industry perspective, that's something that we talk about internally. Is there a path forward that we could be working with regulators and perhaps making the manufacturing of these therapies more readily available for patients?
Yep. So I want to sort of roll forward a little bit, and we discussed the potential for registrational data and filings next year for these initial products. I want to think about beyond the manufacturing side and start to think about infrastructure changes that may be required to actually deliver these innovative therapies, whether they be logistical, reinfusion requirements. Is there anything that you think needs to be put in place from an infrastructure side, almost in the commercial side, that has to sort of catch up with where we are in the clinical, where we are in the manufacturing? And I'll open that up to everybody.
Absolutely. So you can think of this as a therapeutic management system. So what does the company who is manufacturing the drug see as far as the collection of the patient cells? Where is it in the air? When is it getting there? When are the materials for a manufacturer getting there? When is the suite becoming available? When is the testing done, and when does it get delivered? Second element of that is the physician. The physician has got to know they've got a patient. How can they schedule the collection of that patient and the therapy? And then the third is the patient. What does the patient see, right? They're wondering, I donated my cells. I'm waiting for them to be manufactured. I don't know where they are, what's happening, or when I'm going to get them back. And there are interfaces in the system for each of those. And behind that are the logistics of delivery of the collective product and delivery of the final product back. And that's where this crosses over from a drug to a service. And I think my panelists have mentioned that and picking up also from the previous panel, this is so much more of a paradigm shift from small molecules to biologics, as biologics to cells. I don't think people realize how big a paradigm shift this is in the manufacture, the delivery, the reimbursement, and the approvals.
That's true.
This is a big change in how we think about a drug.
And delivering medicine, and ultimately with the goal of a cure. Any other thoughts on the commercial infrastructure side, or are we there yet? Are we behind the 8 ball?
I think like everything else, it's coming together. I mean, we're addressing all of these points. I think the point that I would see as the biggest weakness, so we had, because we've been doing the patient-specific cell therapies predominantly, that's kind of the bulk of our business. We've had about 7,000 products now go to 7,000 patients get products. So that's probably 10,000 products or so that have gone to 7,000 patients. So those touch points coming in and going back to that hospital clinic touch point is where I think the biggest infrastructure need is at the moment. It's just from outside. The thing that I should say, the biggest one that's not really being addressed, because if you're in a big place, if you're in Memorial or if you're at UPenn or if you're at a big center, they know how to handle these things. It's not a big deal, but but you leave that environment very quickly, that next level down, and in handling. And the timing and where the cells are stored and how they're stored. You spend so much time on the manufacturing side, from the time the cells are put in the box till they get back. We spend so much energy and time and money and thought on how to control that. But it's that touch point is a real weakness. It's an Achilles heel at the moment.
One that's critical too, because you could go through all of the manufacturing, and if they're infused improperly or inappropriately.
Absolutely. I mean, people have talked about having the apheresis centers freeze the product, and from a manufacturer standpoint, God, now don't touch it. Right?
Don't freeze it.
Right, but that would add so much shelf life, right? Or not, and that's what you have to be able to control. So it's those control points, I think.
So we're beginning to address that. We have multi-center trials that we have conducted. Novartis and others are conducting multi-center trials. Actually, Novartis has global trials open. So all of those sites that come on first are of the caliber of the academic medical center. But there are others that are coming on that have not been involved in those types of trials before. And this gets to the physician and practitioner education. We have, with our physicians, David Porter, Steve Brupp, and others, been working with the physicians at each of those sites for the multi-center trials to educate them on the collection, but more on the infusion and the management post-infusion.
Yep, makes a lot of sense. So we have just about 3 minutes left, and I've taken more time than I intended to, so I apologize for that. But while we have this great panel, are there any questions from the audience? Yes, please, right up front.
As to the final cost that will be reimbursed, what would you estimate the bag of cells to represent as a percentage of the cost? Because the doctor and the hospital are going to mark up whatever they have.
So it's a question on cost. Sorry, please.
The question is on what's the percent sort of markup of the cost of goods portion of that from a manufacturing standpoint. I hate to answer the question this way, but it's really the only honest answer. It really depends on the product. It depends on how complex the product is to manufacture and what the reimbursement issue comes with. We talked a little bit before about how the field is maturing in this multifaceted approach to solving the issues. It's not just about what the cost is, but it's also about what the reimbursement can be, which will make that formula. And if we can get a value-based reimbursement, it would be great. A lot of people talk about a 30% cost of goods as a number. My sense is it's going to have to be lower than that at the end of the day to really drive the industry, but we've sort of settled in at 30 because it's like, we don't know how we're going to get there at this point. But I think there's a lot of faith for those of us who are tooling around all day that we will get it down, especially in the cases where you can, get it out of the patient-specific realm.
So I think it's interesting. It's a very good question. If you try to find benchmarks such as the pricing of monoclonal antibodies in immunotherapies, then you have to get down to a few $1,000s or max $10,000, $15,000 range of cost of good if you want to keep that barrier of 15% cost of good. I think that sets the goal. If reimbursement can be made at much higher prices, then it's a different ballgame. But we think that at least through the allogenic route, we can get this down to that range and even below that range, keeping space for both margin and also keeping it a cost-effective and accessible therapy, because we believe that otherwise it'll be difficult to broaden its use and make it accessible to a broad range of patients. It'll remain otherwise it'll remain a few patients dealt with in a few hospitals, and so we want this to be global. We need to get this price.
But we may have to start at a higher cost of goods.
It'll start expensive. It'll go down. Yeah. But I think the starting point can be already, antibody-ish.
And we have a question, a gentleman with a microphone right here, please.
Hey, I want to ask about the QA/QC testing part of the manufacturing, and specifically two questions. Number one is, what's the cost of the QA/QC testing for like a per patient batch basis? And then number two, what percentage of cells need to be used for QA/QC testing per production batch?
Okay, I can answer the second question, but not the first. So the percent of cells varies, though, depends on what the dose is that you're generating. We're testing for mycoplasma, endotoxin, sterility, replication-competent lentivirus, and some other testing. But some of those we may not need to do going forward. So if we have a dose that is 1 to 5 times 10 to the 8th, or 2 to 5, or 2 to 8 times 10 to the 6th per kilogram, we have no issue obtaining enough material to do all our QC testing and have backups and have additional reference cells.
Excellent. Well, with that, we're out of time, but I want to thank my panelists for a very, very interesting, very, very lively debate on this important topic. So thank you all very much for joining, and thanks very much, everybody.